Every single second of your life, your body is under attack. Free radicals, unstable, reactive molecules are tearing through your cells, damaging DNA, oxidizing fats in your cell membranes, and corrupting proteins. This is not a metaphor. This is biochemistry, and it never stops. Your body has an answer to this assault, a fat-soluble molecule that positions itself inside your cell membranes, the exact location where the attack begins, and donates an electron to neutralize the free radical before the chain reaction can spread. It sacrifices itself, and then it is regenerated, over and over again. This molecule is vitamin E, and in the 70 years since its discovery, science has revealed that it is not merely an antioxidant. It is a signaling molecule, an immune regulator, a gene expression controller, and a molecule so central to fertility that its original name, tocopherol, literally means to carry a pregnancy in Greek. This is vitamin E, the most misunderstood vitamin in nutritional science, and it is vastly more complex and more important than the supplement aisle has ever told you.
Vitamin E is not a single molecule. It is a family of 8 fat-soluble compounds divided into 2 groups of 4 tocopherols and 4 tocotrienols. Each group contains an alpha, beta, gamma, and delta form. All 8 are antioxidants, but they are not equal and they are not interchangeable. Among the tocopherols, alpha tocopherol is the dominant form, the one the human body preferentially absorbs, retains, and uses. It is the form recognized by the liver's vitamin E transport protein, alpha-TTP, which actively selects alpha tocopherol and packages it into lipoproteins for delivery to tissues. When people say vitamin E, they almost always mean alpha tocopherol. But gamma tocopherol, the dominant form in the American diet, has unique properties alpha tocopherol lacks. It is particularly effective at neutralizing reactive nitrogen species, a specific class of free radical. High supplemental doses of alpha tocopherol actually displace gamma tocopherol from tissues. This is one reason high-dose vitamin E supplementation has shown unexpected results in clinical trials. Vitamin E is fat-soluble. It travels with lipoproteins in the blood and concentrates in cell membranes, adipose tissue, and the liver. Because of its lipid solubility, it positions itself exactly where lipid peroxidation begins, inside the membrane itself, making it uniquely suited to its protective role. Unlike water-soluble antioxidants, vitamin E can reach into the membrane and stop the chain reaction at its source.
The story of vitamin E's discovery begins not with human illness, but with an unexpected problem in rat reproduction. In 1922, at the University of California, Berkeley, Herbert McLean Evans and Catherine Scott Bishop discovered that rats fed a purified diet became infertile. The females could conceive, but pregnancies failed. The fetuses died and were reabsorbed. Evans and Bishop identified a fat-soluble substance in green lettuce and wheat germ that restored fertility. They called it Factor X. In 1924, Bennett Schur named it vitamin E, the next letter available in the vitamin alphabet. In 1936, Evans isolated the pure compound. In 1938, the Swiss chemist Paul Karrer synthesized it and earned the Nobel Prize for his work on vitamins, of which vitamin E synthesis was a central achievement. The name tocopherol was coined by Evans himself, from the Greek tokos, meaning childbirth, and pherein, meaning to carry a pregnancy. The discovery of vitamin E was, at its origin, a discovery about the biological foundations of reproduction. For the next half-century, vitamin E existed in an unusual scientific limbo. It was clearly essential. Vitamin E deficiency caused reproducible, serious pathology in multiple animal species. But defining a clear, unambiguous deficiency disease in otherwise healthy humans proved elusive. This made vitamin E the vitamin that science understood least confidently, and the supplement industry exploited most aggressively. The result was a period of wild overclaiming, and then, when large clinical trials in the 1990s and 2000s failed to confirm those claims, a period of overcorrection and dismissal. The truth, as always, is more nuanced than either camp acknowledged.
Vitamin E operates at the most fundamental level of cell biology, inside the membrane itself. Its functions are broader and more sophisticated than its reputation as a simple antioxidant suggests. Let us work through each one with precision. Membrane protection, stopping the chain reaction. The primary and irreplaceable function of vitamin E is halting lipid peroxidation, the chain reaction destruction of the polyunsaturated fatty acids that make up cell membranes. A free radical steals an electron from a membrane fatty acid. That fatty acid becomes a new radical. It attacks its neighbor. The chain spreads exponentially. Vitamin E donates an electron to break the chain, and is then regenerated by vitamin C in the aqueous phase outside the membrane. Immune regulation. Vitamin E has direct effects on immune function that go beyond protecting immune cell membranes from oxidative damage. It modulates the production of prostaglandins, lipid signaling molecules that regulate inflammation. Specifically, it inhibits phospholipase A2 and cyclooxygenase 2, enzymes central to the pro-inflammatory pathway. This gives vitamin E a genuine anti-inflammatory effect independent of its antioxidant role. In older adults, who show impaired immune function even without frank deficiency, vitamin E supplementation at 200 mg per day has been shown in randomized controlled trials to enhance delayed type hypersensitivity responses, increase antibody titers to hepatitis B and tetanus vaccines, and reduce upper respiratory infection rates. The immune benefit in older adults is one of the most robustly replicated findings in vitamin E research. Gene expression and signaling. Beyond its antioxidant role, vitamin E affects gene expression directly, independent of its electron-donating function. Alpha tocopherol and gamma tocopherol both modulate the activity of protein kinase C, an enzyme central to cell signaling, proliferation, and platelet aggregation. Vitamin E also regulates the expression of genes involved in inflammation, cell adhesion, and extracellular matrix production. Neuroprotection. The brain is the most lipid-rich organ in the body, and therefore the organ most vulnerable to lipid peroxidation. Neuronal membranes are dense with long-chain polyunsaturated fatty acids, particularly DHA, which are exquisitely sensitive to free radical attack. Vitamin E is the primary fat-soluble antioxidant defending these membranes. Severe vitamin E deficiency, seen in patients with fat malabsorption disorders, causes progressive neurological degeneration, peripheral neuropathy, ataxia, and eventually loss of deep tendon reflexes. Eye protection. The retina is exposed to both light-generated free radicals and high metabolic activity, making it one of the highest oxidative stress environments in the body. Vitamin E, in combination with vitamin C, zinc, and copper, has been shown in the landmark AREDS trial, the age-related eye disease study, to reduce the progression of intermediate to advanced age-related macular degeneration by approximately 25%. This is one of the most clinically significant findings in nutritional ophthalmology. Unlike vitamin A, where deficiency is a global health catastrophe affecting hundreds of millions, frank vitamin E deficiency from dietary inadequacy alone is rare in healthy humans. The reason: vitamin E is stored extensively in adipose tissue, and the threshold for depletion is high. But this does not mean adequate vitamin E status is universal. It is not. Primary dietary deficiency does occur, most severely in populations consuming very low-fat diets, where fat-soluble vitamin absorption is impaired. Secondary deficiency, the more clinically significant form, occurs in patients with fat malabsorption disorders, cystic fibrosis, Crohn's disease, short bowel syndrome, cholestatic liver disease, and abetalipoproteinemia, a rare genetic condition in which lipoproteins cannot be produced and vitamin E cannot be transported. The neurological consequences of severe vitamin E deficiency are among the most striking in nutritional medicine. Patients with abetalipoproteinemia develop a progressive spinocerebellar syndrome, ataxia, peripheral neuropathy, loss of proprioception, and retinal degeneration that closely resembles a genetic neurodegenerative disease. These symptoms can be arrested and partially reversed with aggressive vitamin E supplementation. Toxicity. Vitamin E is among the least toxic of the fat-soluble vitamins. Unlike vitamin A, where toxicity is a significant and well-documented clinical concern, vitamin E toxicity from food is essentially unknown. However, high-dose supplementation, above 1,000 milligrams per day, has raised two specific concerns in the literature. First, vitamin E has anticoagulant properties. It inhibits platelet aggregation and antagonizes vitamin K-dependent clotting factors at high doses. Patients on warfarin or other anticoagulants who take high-dose vitamin E supplements can experience bleeding complications. Second, the 2005 meta-analysis by Miller and colleagues, widely cited as showing that high-dose vitamin E increases all-cause mortality, has been substantially criticized for methodological flaws, including the inclusion of studies using doses far above established upper limits in already ill populations. The current tolerable upper intake level for supplemental vitamin E is 1,000 milligrams per day for adults. Below this level, evidence of harm in healthy individuals is limited. The practical message: do not fear dietary vitamin E, but do not treat high-dose supplements as automatically beneficial. Where is vitamin E found in food? The answer is more specific and more surprising than most people expect. Vitamin E is concentrated in plant oils, nuts, seeds, and a small number of exceptionally nutritious whole foods. Understanding which sources provide the most bioavailable forms is essential. The single richest source of alpha-tocopherol, the most biologically active form, is wheat germ oil. 1 tablespoon provides over 20 milligrams of vitamin E, more than the entire recommended daily intake for an adult, which is 15 milligrams. Sunflower oil and safflower oil are next, high in alpha-tocopherol. Olive oil is moderate. Palm oil contains significant tocotrienols, a different, increasingly researched form. Nuts and seeds are outstanding sources. Almonds lead. A 30-gram handful provides approximately 7.3 milligrams of alpha-tocopherol, nearly half the daily requirement. Sunflower seeds are exceptional. 30 grams provides over 7 milligrams. Hazelnuts, pine nuts, and peanuts follow. Brazil nuts, famous for selenium, also provide meaningful vitamin E. Avocado is one of the most nutritionally significant whole food sources. A medium avocado provides approximately 4 milligrams of vitamin E alongside its monounsaturated fats, which also enhance absorption. Spinach and other dark leafy greens provide meaningful amounts. 1 cup of cooked spinach provides approximately 4 milligrams. Red bell peppers, asparagus, and broccoli round out the vegetable sources. The critical practical point: because vitamin E is fat-soluble, it is absorbed alongside dietary fat. Eating spinach or bell peppers alone provides vitamin E, but adding olive oil or eating it alongside fat dramatically increases absorption. The most vitamin E-rich meal is one that combines nuts, seeds, or oils with colorful vegetables in a fat-inclusive preparation. One underappreciated point: cooking and food processing destroy vitamin E. Refined vegetable oils have significantly lower vitamin E than cold-pressed versions. Roasting nuts at high temperatures degrades tocopherols. Frying with oils at high heat accelerates vitamin E depletion. Choosing minimally processed, cold-pressed oils and lightly prepared nuts and seeds preserves dramatically more of this heat-sensitive vitamin.
Everything we have covered, now made completely actionable. Here is what the science actually supports for food, supplementation, and daily practice. Eat a small handful of almonds or sunflower seeds every day. This single habit provides nearly half your daily vitamin E requirement in its most bioavailable form, alpha-tocopherol, embedded in a food matrix with natural fat for absorption. Almonds also provide magnesium, calcium, and protein. Sunflower seeds deliver selenium alongside vitamin E. The combination is nutritionally powerful. Use cold-pressed oils and add fat to colorful vegetables.
Drizzle cold-pressed sunflower, safflower, or olive oil over spinach, bell peppers, and broccoli. This simultaneously delivers vitamin E from the oil and dramatically increases the absorption of vitamin E already present in the vegetables. This is the highest impact dietary vitamin E strategy available from everyday cooking. Eat avocado regularly. A medium avocado provides nearly a third of your daily vitamin E alongside the monounsaturated fat that ensures its absorption. It also provides potassium, folate, and fiber. Half an avocado at lunch or dinner is one of the most nutritionally efficient habits in preventive nutrition. Supplement with caution. Choose mixed tocopherols, not alpha alone. If you supplement, choose a product containing mixed tocopherols, alpha, gamma, and delta forms, rather than synthetic DL-alpha tocopherol alone. High-dose alpha tocopherol displaces gamma tocopherol from tissues, potentially losing the unique benefits of the gamma form. Stay below 400 milligrams per day unless under medical supervision. If you are on anticoagulant medication, discuss vitamin E supplementation with your physician first. We began inside a cell membrane, watching free radicals tear through lipid layers that had no defense. We followed the tocopherol molecule as it positioned itself at the exact location of attack, donated its electron, and ended the chain reaction before it could spread. We traced its discovery from a rat colony in Berkeley to a Nobel Prize in Zurich. We saw it operating inside the nervous system, the retina, the immune cell, and the aging brain. We saw the clinical trials, the ones that confirmed its benefits and the ones that complicated the story. We saw the nuance that the supplement aisle never told you. Vitamin E guards every cell. In the literal sense, through the tocopherol embedded in every membrane of your body, and in the broader sense, as a molecule that has been quietly, invisibly, continuously protecting the biochemical integrity of your tissues since the moment your cells first formed. If this video showed you something about a molecule you thought you already understood, share it with someone who deserves to know. Subscribe for more evidence-based explorations of the molecules that determine your health. And tell us in the comments, which vitamin E food source will you add to your diet this week?